Okay, so the goal of lipolysis is to try to convert a triglyceride molecule into an ACOA molecule, because those ACOA molecules can then be used in the Krebs cycle to make more ATP. And then once we have that ATP, we can contract our muscles. So the first step is going to be on the left-hand side under that M, which is mobilization. So whenever we exercise, hormones are released into our bloodstream, and then those hormones can bind to the triglyceride molecule. And then a hormone-sensitive lipase breaks apart that glycerol backbone from those fatty acid chains. So now those fatty acid chains are free, and they can move into the blood, which is shown by that blue arrow. So now we can move to the second step, which is transport and cellular uptake, which is that first T. So now the fatty acid chains are in the blood, and they bind to that black circle, which is albumin. And then the sarcolemma fatty acid binding protein, which is that blue triangle, allows for those fatty acids chains to be moved from the blood into the muscle. But it only allows for about 50% of these now free fatty acids to move into the muscle, and it doesn't take up 100% of all of the fatty acids, because other places in your body besides that muscle need that fat. And then the third step is activation. So now we're in the cytosol of that skeletal muscle cell. So we have a free fatty acid and then a coenzyme A, which is that CoA, and that CoA just helps the enzyme do its job basically. And then on that arrow, we're going to have ATP convert into ADP through dephosphorylation. And you're also going to have an ACOA synthase. So those two things are going to help FFA and coenzyme A convert into a fatty acid ACOA, which is just FACOA, as we see later. Okay, and then we can move to step 4. So this is translocation. So all we're doing is moving it from the cytosol to the mitochondria, but in order to do that, we need that blue triangle, which is just carnitine transferase, and that's it. So then we go to step 5, which is oxidation. And the goal here is to just cut up that FACOA that we just made into ACOA, because that ACOA can be used in the Krebs cycle to get more ATP, which is what we need for muscle contraction. So in this example, our FACOA molecule only has 6 carbons, but it can have more. And so it's going to undergo beta oxidation, which just cleaves 2 of the carbon molecules and combines it with an ACOA molecule. And then you'll have 4 carbons still left over. So in order to get those down to 2 carbons, to go with another ACOA, which is the goal, you'll have to go through beta oxidation again. So in this case, that's our last step, because we'll have 2 carbon molecules and an ACOA, and then another 2 carbon molecule and an ACOA. And there's nothing more to break down. So those 3 ACOA molecules can go into the Krebs cycle to now be used for ATP production and muscle contraction.